Okay, so good afternoon, everyone. Welcome to Medical Grand Rounds. Before we get started, I'd like to make a brief land acknowledgement. McGill University is on land which has long served as a site of meeting and exchange amongst Indigenous peoples. We acknowledge and thank the diverse Indigenous peoples whose presence marks this territory on which people of the world now gather. We are very fortunate tonight to have a guest speaker coming to us courtesy of the Division of Endocrinology, and I'd like to ask Dr. George Ivan-Fantis to present our guest speaker today. George?
Okay, thanks, Nadia. Welcome to everyone in our hybrid in the room and also those online. So it's a pleasure today to introduce our speaker. Before I do that, I'd like to just state that this is an annual Novo Nordisk Lectureship, which is the most prestigious diabetes lectureship at McGill. And I'd like to thank Novo Nordisk for their long standing funding of this and support of this lectureship. This is, in fact, our 40th year. So it's a very long tradition. There are a few people, a list of Novo Nordisk people, but I'll save some of the specifics for this evening's lecture. Our guest, Dr. Myers, will be giving a lecture also this evening as part of this event, of this lectureship. And we'll thank those involved in more detail. So Dr. Martin Myers is a professor of internal medicine and molecular integrative physiology in the Division of Metabolism, Endocrinology and Diabetes at the University of Michigan Health System. He holds the Marilyn Vincent Professorship of Diabetes Research. Dr. Myers completed his undergraduate degree at Princeton University and his MD and PhD at Harvard Medical School. He did that combined program that we also have at McGill, and of course went on to a fantastic research career after that. He undertook a fellowship at the Joslin Diabetes Center in Cellular and Molecular Physiology and was appointed to the faculty of Harvard. His talent was quickly recognized and he was recruited to Michigan, where he became director of the Michigan Diabetes Research Center. And as you see on the slide, he founded and developed the Elizabeth Weiser Caswell Diabetes Institute there. His early work was on the molecular mechanism of insulin signaling, but when he moved, his major focus switched to leptin signaling, obesity, metabolic syndrome, and the hypothalamic and central nervous system control of appetite and energy balance. This has been well funded over many years, and he's internationally renowned leader in this field. His high-impact research has been recognized by numerous awards. I'm just going to mention a few to save time. The Jerome Award for Research Excellence, an NIH Merit Award, 2 Outstanding Scientist Awards, one from the Obesity Society, another from the American Diabetes Association, the Ernst Oppenheimer Award of the Endocrine Society. He's been elected to the American Society for Clinical Investigation and the American Association of Physicians, and received a fellowship in the American Association for the Advancement of Science. He serves on many grants panels, editor and editorial board of major journals, and in fact, he co-founded the journal Molecular Metabolism. He teaches a lot of students and mentors postdoctoral fellows who've gone on to their own careers. His work on the intricacies of leptin signaling via the CNS has shaped our understanding of the pathogenesis of obesity and type 2 diabetes. So it's a pleasure to welcome Dr. Myers as this year's 2024 McGill Novo Nordisk Lecturer. Dr. Myers.
So thank you, Dr. Fantis, and thank you for not just for that introduction, but for having me here. It's an absolute delight, a beautiful city to visit. And of course, there are a wonderful bunch of scientists here that I already know that are just a delight to interact with. As Dr. Fantis said, I'm giving 2 lectures today. This is the Medical Grand Rounds version, where I'm going to try to sell you on the fact that diabetes is bad, that obesity causes diabetes, and then that there are systems in the brain that actually tell us how much we should eat, when we should stop eating, and at the end of the day, dictate how much we eat and how much we weigh. And then I'm going to talk about ways that we are trying to, we, meaning the field at large, not just me, therapeutically intervene in those systems and where probably the best places to intervene are going forward. Okay, so with that having been said, again, I'm going to tell you some of the things that you already know. Diabetes in Canada is exploding. As of 2016, there were a little over 3 million people in Canada with diabetes. That's an effective rate of about 8% of the population. If it's anything like the United States, I'm sure it has gone up from there in the past 8 years. There are a bunch of risk factors for type 2 diabetes. Again, all of you know this, so I'm going to go over this quickly. Obviously, genetics, family history matters. There are a lot of environmental factors, inactivity, weight gain, age. Of course, most diabetes is type 2 diabetes, and so as we get older, we have a tendency to get type 2 diabetes. People with polycystic ovary syndrome or who have gestational diabetes are at a higher risk for developing type 2 diabetes. And then race and ethnicity matters. So African ancestry, people of Hispanic origin, people of indigenous origin, and people from Asia actually have a much higher incidence of type 2 diabetes than do people who are from Western European descent. Now, of these risk factors, as you might guess, the one I'm going to focus on today is weight gain. And in fact, it's weight gain that's really, and obesity that's really driving the explosion of diabetes, type 2 diabetes, both in Canada and in the United States. So these are slightly more recent data, and you can see the percent of adults in Canada living with obesity from 1970, where it was somewhere around 7 or 8% to currently, where for both men and women, it's hovering around 25%. If there's good news in this slide, it's that the rate of obesity seems to be leveling off a little bit. And that's, I guess, because we can only get so fat before everybody is fat. If you look at not just obesity, but overweight in Canada, and you look at the age prevalence, once you're over 35, you have between a 60 and 70% chance of being either overweight or obese. So there are a lot of things that are pushing us. I feel guilty saying this while people are eating lunch, but there are a lot of things that are pushing in the direction of increasing type 2 diabetes. So what I'd like to start by trying to convince you today is that body weight, food intake is regulated. In fact, it's regulated in a manner similar to blood pressure. And I'm trying to get this point across because a lot of times, not only in lay audiences, but in well-educated medical audiences, people think, somebody who's obese, they should just stop eating so much. And while that's true, it actually turns out to be very difficult because the way these systems in the body work, if you look at this graph, and this is not real data, this is just a theoretical thing. If you look at the amount of food people eat over 24 hours, and compare that to their body mass index. We've got an idealized person who's got a body mass index of 25, which we know no longer actually represents reality. If you take that person at their sort of steady state weight and you ask them to diet, and so that decreases their body mass index, that does two things. One of them is that it increases hunger. The other is that it decreases energy expenditure. And both of those together tend to push weight right back to where it started. Now, it actually turns out that it's regulated in the other direction too. If you overeat, maybe it's the holidays or something like that, your body mass index increases, your appetite actually tends to decrease and your energy expenditure goes up. And that again pushes you back in the direction of homeostasis where you started. And I just want to give you some evidence that this system exists. There is evidence actually from humans, but because those studies were done on imprisoned people, we tend not to show those anymore. So I'm showing you rats. In this population of rats, you can see they're eating along. They start at about 250 grams, and the control rats are just sort of eating along and gaining weight. The reason that they continue to gain weight is actually rats, unlike us, continue to grow linearly as well as width-wise throughout their lifespan. If you take a similar set of rats and you decrease their food intake to the point where a few months later, their body mass is down by about 15%. If you keep them on that for, say, 2 or 3 months, and then all of a sudden you let them eat ad libitum, you can see what happens. They start eating like crazy. I'm not showing you the food intake data, but essentially their body mass bounces right back to where it was before. Now, you may notice that there's a gap here between the controls and these guys, and that's because the food-restricted animals actually stopped growing a little bit. And so even though they came back to parallel the original curve, they didn't quite get back there just because they don't have quite as much lean mass as the controls. And again, this works in the opposite direction. So if you take rats and you force overfeed them, which they don't like, so you actually have to pump food into their stomach to do this. Nobody likes being overfed. If you overfeed them for about 3 months and you keep them overfed for another month and a half or so, and then let them go back to ad libitum feeding, their food actually over the first couple days plummets to 0. And then over the time of about 2 or 3 weeks, their body mass comes back down and then follows the line of the control rats from that point on. So there is a system in the brain that tells you how much you should weigh, how much you should eat, and tries to keep things homeostatic. Now, this system is actually really, really accurate. If you just look at humans, so the average human energy intake per year is close to 1 million calories. The average human also gains about 1 pound, so half a kilogram a year. That's about 4,000 calories. So that's an error rate of about 0.4%, which is pretty small. And if you think about that sort of in absolute calorie terms, that's 11 calories a day. That's a lifesavers, right? It's a little one of those lifesavers candies. So the system really does do a pretty good job, and you only need to be a little bit off from the target that your brain is setting for you to gain weight. The other thing that I'll point out is that body weight regulation is not like a thermostat. It's not like the system in your brain says, okay, Paul Sabatini here should weigh, exactly 165 pounds. The way it works is there's a settling point, and there are a whole bunch of things that go into that. So if you think about pressures on the control of food intake, so there's obviously genetics. We talked about that, and you can have a genetic background that actually makes you more or less prone to eat more and gain weight. You can have decreased fat reserves. So this is what happens when you go on a diet, right? When you go on a diet, your fat reserves go down. That decreases leptin levels, and we'll talk a little bit about leptin in a minute. And that tends to make you hungry and push up on food intake. Similarly, if your body perceives that you are burning more energy than you usually do, and that's because you ramped up your exercise or you went and sat in the cold or something like that, your body perceives that you're going to burn more calories and says you need to eat more calories, and that pushes up on food intake. Also, easy to get tasty calories. So in the world that we live in, it's very easy and very inexpensive to find calories that are really delightful to consume. And if you compare that to back in the day when everybody was eating essentially whole grains that they then had to grind and prep themselves, it's a completely different world, and it's much easier to ingest all those calories. So obviously the opposite is true. Increased fat reserves tends to push down on food intake. And obviously when it's harder to get less tasty food, that pushes down on food intake as well. So if you think about what I've just said, what I've basically just said is that, back in the day, before we had all of these lovely, readily available calories, if you take a body mass index of 30, most people lived below that body mass index of 30. Now, there are a bunch of people above it, depending on where you are in the world, somewhere between 30, 40%. And there are 2 reasons for that, right? Obviously, genetics didn't change between then and now. What changed is our environment. We have all of this plentiful food, we have less need to exercise, so on and so forth. And you can see the same thing in mice, right? You take C57 black 6 mice, which are very diet sensitive, you put them on a high-fat diet, and over about 12 weeks, their body fat goes up by about 2.5-fold. Now, so that's the environment effect, but it turns out, as for humans, the genetics of these mice affects how they respond to that that diet. So FVB mice, which are actually more closely related to each other than all of the people in this room are to each other, unless they're brothers and sisters here, they don't actually gain much weight on a high-fat diet. So let's start talking about the CNS systems that control food intake and body weight. This is actually a figure that I cribbed from a publication from Mike Schwartz from, oh gosh, close to 20 years ago at this point. And what he's showing you here is a system where the fat cells in your body make a hormone called leptin. And the more fat you have in those fat cells, the more leptin they make. And that leptin goes through the circulation, winds up binding to the leptin receptor in the brain and saying, hey, this is how much energy you have in your body, and you should behave thusly as a consequence. And when I say behave, that's not just the behaviors like food intake, but as we'll talk about in a minute, in terms of a variety of things that control and are affected by energy balance. And we think of the hypothalamus as the place that integrates all of these signals of energy stores and a variety of other signals, and they modulate body weight and food intake, not necessarily instantaneously, but over the long term. These are the systems that say your body weight is, your fat mass is thus now, and it should be the same, 2 weeks from now or 2 months from now. Then there's this other system, and what Mike is showing is signals going in from the gut to the through the vagus nerve up to the brainstem and the nucleus of the solitary tract. There are gut peptides, of course, that are elaborated in response to feeding too. These are signals of feeding that are nutrients in the gut, stretch of the gut and intestine wall, and they go up to the brainstem. So the job of the brainstem, at least in terms of classical thinking, is that it receives information from the gut and some other acute signals to limit food intake over the short term. And that prevents us overfeeding and ingesting more calories than we can safely store in the short term. It also, in some cases, prevents us from the continued ingestion of toxins if we eat something that is not good for us. This part of the brain actually contains the system that will help us expel them and never eat those things again. And I'll talk more about that as well. So I'm going to say a little bit more about leptin. So leptin is encoded by the OB or the obese gene. It was cloned by Jeff Friedman in 1994. What I'm showing you here is C57BL6 OB animals. So these are mice that don't have leptin. Those of you that don't do a lot with mice won't know this, but a 60-gram mouse is about two and a quarter times as big as your ordinary sort of wild type mouse. And you can see that if you just sort of let them go along eating on their merry way, they're eating a ton of food, their body weight is staying stable or creeping up just a little bit. But if you treat them with leptin, which is what the black line is here at the bottom, you can see that food intake goes down by about 70%, and their body weight starts going down, and even a month later, it's continuing to go down as they trend towards normal. You can also take wild type animals, these are lean wild type animals, and you can give them leptin, and you can see that food intake will actually go down, not as dramatically, but they're not eating as much as the OBs were. Eventually, food intake comes up pretty close to where they were at baseline, and you get a decrease in body weight as well, but the decrease in body weight doesn't go down as much as the really leptin sensitive, leptin deficient animals. It only goes down by maybe say 5 or 10%. And then you take DB animals. DB animals are actually missing the leptin receptor. And so they start up at about the same body weight as the leptin mutant animals, and they're eating about the same amount as the leptin mutant animals. But if you give them leptin, of course, nothing happens. They have classic leptin resistance, right? There is no receptor, the hormone can't do anything, so on and so forth. Now, leptin doesn't just control food intake. I think that's an important, food intake obviously is an important thing that leptin does control, and it's something that everybody thinks about a lot. But if you think about leptin in evolutionary terms, leptin was probably not evolved, if you want to think about it that way, to prevent us from overeating. Because if you're a hunter-gatherer on the Serengeti, it's very unlikely that overeating is a problem that you're going to experience very often. The more likely problem you're going to have is that you haven't had an antelope in a while. And so as you're wandering around looking for antelope, being very hungry because your leptin levels are low if you haven't had that antelope in a while, you also need to hold on to the calories that you have. And so it turns out that the fall in leptin levels is what causes what we think of as the neuroendocrine starvation response. So I'm showing you data here from Rex Ahima and Jeff Flyer from a bunch of years ago. These are leptin levels in fed animals. They took a bunch of animals and they fasted them for 48 hours, which is actually a relatively extreme fast for mice. And then they had a separate set of animals that were fasted, but they gave them exogenous leptin back to bring their levels back up to normal. And what I want to point out here is that if you look at their hormone status, so for instance, thyroid hormone, the fed animals are at about 0.85, the fasted animals are plummeting down to about 25% of that, and you can bring that back, not all the way, but at least part of the way by giving them leptin. Same thing for corticosterone and ACTH. Starvation is a pretty strong stress, so starving animals, the corticosterone goes up. You can push that back down by providing exogenous leptin. And then, of course, the other thing that leptin controls is the reproductive cycle, because reproduction, especially in females, is extraordinarily energetically costly. When you think about the cost of actually growing a fetus inside you and then having to lactate afterwards and provide all of those calories for the growth of the animal, if you don't have enough calories on board, reproducing is definitely something that you want to do. So when Jeff and Rex did this experiment in female animals, they actually followed estrus, so essentially ovulation after that. And you can see the normal mouse has an estrus cycle every 3 to 5 days. The animals that were fasted, and even they were only fasted 48 hours before they let go, it took them about 10 days before they had their next estrus cycle, whereas the ones that got leptin came back in about 3.5 or 4 days. So leptin is really the fall in leptin levels is the important signal that you need to turn on this thing called the neuroendocrine starvation response, and hold on to the calories that you have in any way that you can. This is actually true in humans, not just in mice. So now I'm showing you data that came from Rudy Leibel and Mike Rosenbaum at Columbia. Again, these are not new data, but I think they're probably the best example we have of how all this stuff works in humans. What they did is they found a bunch of people in New York City who are willing to be admitted to the hospital for 2 months, during which they sort of kept them weight stable for a little while, and then they reduced their food intake over a few weeks so that at the end of the day, these people were about 10% lower in body weight than they were when they initially came in. And they studied them at that point, and then they kept them at that weight and they gave them leptin and looked to see what happened comparing the 10% weight loss versus 10% weight loss with leptin. And this is just to show you that when they do this, the people whose body weight was down 10% obviously had lower leptin levels, and they were able to normalize leptin with the provision of exogenous leptin. And if you look at their energy expenditure, you can see the 10% weight reduced people had a change in total energy expenditure of about 20%. That was restored pretty close to normal by leptin treatment. And then I'm just going to tell you that they did the same thing with thyroid hormone and a bunch of other things. But then the other thing they did is actually ask them questions about how they felt after a meal. And this is a relatively arbitrary scale, but you can see that a normal person after a test meal has a sense of fullness that they rate at a 110. The people who were 10% weight reduced had a 93, so they felt like they weren't as full. But the people who got leptin actually felt like they weren't as hungry as the people who were similarly weight reduced. And then when you asked them to rate how much they had eaten, because of course this was a defined test meal, the people who were 10% weight reduced felt like they had eaten about 10% less than they actually had, and that was restored by leptin as well. So these systems don't just exist in animals, they exist in us, and they do similar things in humans as they do in animals. So let's talk for a minute about the hypothalamic circuitry that mediates these effects. Now, what I'm going to show you is a very simplified diagram of how all this works, but it is, in fact, the nuts and bolts. So the main system in the hypothalamus that modulates a lot of this stuff is what we call the hypothalamic melanocortin system. This is a set of neurons in the arcuate nucleus that express pro-opium melanocortin, which gets chopped into alpha MSH, and alpha MSH from the POMC neurons gets released onto melanocortin 4 receptor expressing cells in the paraventricular hypothalamic nucleus. And that decreases feeding. It also has some effects on energy expenditure that we won't necessarily get into today. There are leptin receptors on the POMC neurons. Leptin binding to leptin receptors increases the expression of pro-opium melanocortin, et cetera, et cetera. There are also a set of neurons in the arcuate nucleus that we think of as competing. These contain AGRP and NPY. They release both of these inhibitory neurotransmitters plus the inhibitory neurotransmitter GABA. They project onto a lot of the same cells in the paraventricular hypothalamus that the POMC neurons do, but they, of course, inhibit those cells. So when an AGRP NPY neuron is on, it's firing, it's actually increasing food intake. And as you might imagine, these neurons also have leptin receptors. Leptin acts on these and actually inhibits the AGRP neurons. Now, it turns out actually that it's a little bit more complicated than that, even though this is a nice simple model. It turns out that a lot of what leptin does to AGRP neurons isn't via direct leptin action on the AGRP neurons, but indirectly by activating a set of neurons in the place called the DMH that then inhibits the AGRP neurons. But we don't need to go into those details. So what about targeting this system, which is in fact the system that we use to control long-term energy balance in the brain therapeutically? Well, everybody, of course, upon the cloning of leptin thought that this was a really good idea. And I'm just going to show you that it doesn't actually work as well as we thought. So this is the first hint that there was something going wrong here. Or actually, if you think about it, going right. So it turns out if you look at leptin levels and you compare that to total body lipids, so essentially adiposity, the heavier you are, the more leptin you have. And that makes sense, right? I already told you that the more fat you have, the more leptin you make, because fat cells make leptin, and the more fat they have, the more leptin they make. But this also suggests that if you want to use leptin as a therapeutic, it's going to be a little bit of a problem, because these animals theoretically aren't responding appropriately, and I'm putting appropriately in air quotes, right? They're not responding the way we'd want them to the excess leptin that they're already generating. And in fact, if you take lean animals and you put them on a high-fat diet to get them to start gaining weight for 4 days, 16 days, and 56 days, you can see that leptin-treated animals after 4 days on high-fat diet you can do a pretty good job of suppressing their food intake. They're still suppressing their food intake a bit in response to leptin after 16 days, but after 56 days, the leptin's not doing anything anymore. So people call this leptin resistance. I'm fine with us calling it leptin resistance today. If anybody actually wants to talk about why I hate that term, we can talk about that later, but that I think is a story for a different day. The bottom line is animals that are obese and have lots of leptin already, it doesn't help if you try to give them more leptin. And that's not, again, just true in animals, it's true in humans. This is an early study where they took pegylated leptin, they gave it to a bunch of obese people, and you can see that leptin levels go up initially very high, and then, as they reach steady state, leptin levels are up about, 20–30% compared to where they were at the starting point in these humans. And what happens to food intake and body weight? I'm not showing you the food intake data, but if you look at the placebo-controlled people, they lost about 6.4 kilograms, so about 2% of their body fat. The folks that got leptin actually did worse. Not statistically significantly worse, but they functionally didn't lose quite as much weight. They didn't lose quite as much fat mass as the controls did. So giving leptin to obese people in an attempt to get them to lose weight isn't going to work. Presumably, in my opinion, because they're already saturated. There's a maximum of what you can get leptin to do, and they're probably already there. So that's not to say that leptin is completely therapeutically useless, because there are actually humans out there who don't have leptin for a variety of reasons. So there are very rare cases where there are genetic deficiencies in leptin. This was the first one of those people who was identified by Sadaf Faruqi at Cambridge in the UK. And you can see this is the body weight of this person that she was following. She started leptin therapy on this person here, and you can see body weight started going, BMI started going down here. You can follow it out for 12 months here. You can see that their body mass goes down like crazy, but it's fat mass, not lean mass, that they're losing. And the other thing is this person turned out to be a female. And of course, they were amenorrheic. But once they started getting leptin, it turns out they started having estrous cycles. Similarly, if you take people who have hypothalamic anorexia, these are often people who have eating disorders or who are highly trained athletes, have very low levels of body fat and consequently have very low levels of leptin. This is a study by Chris Montouros at the BI Deaconess, and you can see some of his patients had basically 0 LH pulses at baseline. When he gave them 2 weeks of human metroleptin, he was able to restore their pulsatility, and they actually began menstruating again. Obviously, he was giving them leptin, so they didn't start eating more. They didn't gain any body mass, any fat mass, or anything like that. Now, I don't think anybody's suggesting that you should use leptin as therapy for hypothalamic amenorrhea. That's a different thing. But there is actually one other class of patients, again, relatively rare, who benefit from leptin therapy. And these are folks with lipodystrophy. So lipodystrophy, there are a variety of flavors. There are many congenital, some acquired, some partial, some complete. But the bottom line is these folks have very little fat mass. And as a consequence, they make almost no leptin. As a consequence of having no leptin, they're hungry and they eat a lot. And they have all of the effects that you would expect from leptin deficiency in females. They have hypothalamic amenorrhea, but also because they have nowhere to store the calories that they're eating in terms of adipocytes, they tend to shove their calories into their liver. They have extraordinarily high triglycerides, as you can see for this person here with high triglycerides at about 10,000 milligrams per deciliter, and they're getting plasmapheresis. Their glycated hemoglobin was pretty high, and of course the serum leptin was undetectable. This is a sort of a CT image of what that person's abdomen looked like, and you can see there's a big fatty liver there in the middle of it. And I should say that this actually was a study done by Elif Oral, who was at the NIH at the time, but who's now at the University of Michigan and runs a big lipodystrophy clinic even now. So what Elif did was to give these folks leptin, and you can see this first person who got leptin, after about 3 months in leptin, triglycerides were down close to normal. Glycated hemoglobin started coming down. Obviously, you could detect leptin, and the size of that liver went way, way, way down. So leptin's not a great therapy for obesity, but it's really good for people who don't have leptin. So I've told you that leptin's not a great therapy for people who have garden variety obesity. What about the melanocortin system? Well, I'm going to tell you, but not show you, that there were a bunch of melanocortin 4 receptor agonists that people tried to push into the clinic probably 20, 25 years ago. Most of these had unwanted side effects like big increases in blood pressure, and they actually also had modest weight loss efficacy. But as with leptin, it turns out that there are some uses for these things. They're not necessarily garden variety uses, but they're out there. So this is a relatively new MC4 receptor agonist called setmelanotide, and it treats patients that have disruptions in this sort of hypothalamic melanocortin system that are upstream from the MC4 receptor. So these are a couple patients who have a very rare mutation in pro-opiomelanocortin, so they're not making any alpha MSH. They obviously have disruption of the hypothalamic pituitary adrenal axis as well that you can't cure with setmelanotide, but if you take these very obese people that have no pro-opiomelanocortin and you give them setmelanotide, you can see their body weight comes down pretty rapidly. It's also true of people who have mutations in the leptin receptor, right? You can't treat people who have mutations in the leptin receptor with leptin because they are completely and utterly leptin unresponsive. But because a part of the problem that they have because of leptin deficiency is because they don't have enough tone in the hypothalamic melanocortin system, if you give them setmelanotide, you can get their body weight to come down. They also, become less hungry, and there are a lot of other metabolically beneficial side or effects, but I'm not going to go through all of those today. Again, these are very, very, very rare people. Okay, so let's think about whether there are other ways that we could potentially target the melanocortin circuitry. Turns out the POMC neurons contain or express the serotonin 2C receptor, and serotonin 2C receptor is the receptor for a class of serotonin 2C receptor agonists, including olorcaserin. So it turns out, and this is some basic science data, that if you take a serotonin receptor 2C null mouse, so no serotonin 2C receptor anywhere in the animal, you give them lorcaserin, and you don't get any decrease in food intake, not surprisingly. Their baseline food intake is actually quite high as well. But if you restore serotonin receptor 2C expression just in the POMC neurons, now lorcaserin works. And of course, these animals are lighter at baseline. So actually, lorcaserin and other serotonin 2C receptor agonists work via POMC neurons. This is actually an approved drug by the FDA, and you can give it to humans and they will lose weight. This is showing what happens after about 3 months. And so this is better than anything I think we had previously. Now, the bottom line, though, is that after 3 months, these patients have only lost about 3 or 4 kilos. And at that point in time, they've plateaued. So this isn't enough efficacy to treat all of the people and the magnitude of obesity that we have out there. GLP-1 receptor agonists, I think everybody has heard about them at this point in time. They do much better. Right here, I'm showing you mouse data. You take mice that are sort of eating along, you give them semaglutide, which is a GLP-1 receptor agonist that's a once weekly injectable, injected daily in mice, but that's a different story. And you can see that after about 7 days, 1 week, these animals have lost somewhere around 20% of their body weight. It works in humans too. This is an early study. And just showing you that not only does semaglutide, which was originally prescribed for the diabetes indication, decrease fasting plasma blood glucose, but it causes changes in body weight. And if you look at the change in body weight over 12 weeks, you can see that these folks have actually lost somewhere in the range of 4.5 or 5 kilos, and they're still on their way down if they're on the highest dose. So this stuff looks like it's doing much, much better. So this has got to act in the hypothalamus, right? Because we're changing long-term food intake and body weight. So what I'm showing you now we're back into the mouse data from Randy Seeley. What Randy Seeley did is he knocked out the GLP-1 receptor specifically in the brain of mice and then asked whether they could respond to liraglutide. And it turns out they can't, right? You give them liraglutide, no decrease in body weight, and that's matched by no decrease in food intake. So then he went on and did the same thing looking at the hypothalamus. So basically what you have in each of these graphs is 2 lines looking at the response to lira in animals that are either wild type or that are missing GLP-1 receptor throughout the hypothalamus. And what you can see here is that you don't need GLP-1 receptor in the hypothalamus to lose weight in response to GLP-1 receptor agonists. In fact, you don't need it in the POMC neurons in the hypothalamus, and you don't need it in a different part of the hypothalamus called the paraventricular hypothalamus. So coming back here, how do we make sense of this? Right. So I told you that the hypothalamus is the place that's really important for the control of long-term control of food intake and body weight. And I've shown you that when we target the hypothalamus, we don't do a great job of decreasing body weight, at least with the ways that we've tried so far. And I've also showed you that there's this new great set of weight loss drugs called GLP-1 receptor agonists, and I've shown you that they don't work in the hypothalamus. They probably actually work in the brainstem, and that's where we're going to be for the rest of this talk. So I'd like to start by talking about how the gut signals to the brain. So because we need to know a little bit about that to sort of get into the weeds here for a minute. So the gut, when you ingest food, secretes peptides that go through the circulation. Those peptides bind to neurons in a place called the area postrema, which actually lives outside the blood-brain barrier, so it's really good at sampling the blood. The area postrema then pushes those signals forward into a place called the nucleus of the solitary tract. Nucleus of the solitary tract also receives some input from circulating gut peptides directly. And then there's a bunch of information that comes in via the vagus nerve, right? They're nerves that are sensory. They sense stretch and nutrients in the gut. And all of those neurons synapse in the nucleus of the solitary tract as their first step. And somehow all of these signals from the gut send a signal that says, stop eating, right? Satiation, you've eaten enough calories and it's time to stop. So it turns out that there are GLP-1 receptor containing neurons both in the area postrema and in the nucleus of the solitary tract. So the question is, what do they each do? Can we identify one or the other population that's more important for the control of food intake, thinking that we might want to target it? So before I get into the circuitry, I'm going to tell you about the tool that we use for a lot of these things. These are called designer receptors, exclusively activated by designer drugs, DREADs. These are essentially human muscarinic receptors that have been mutated so that they no longer bind acetylcholine. Their ligand is this otherwise inert compound called clozapine N oxide. So if we put these DREADs in a neuron, we can actually turn on the neuron by giving the animals clozapine N oxide. But the clozapine N oxide doesn't have effects anywhere else in the body. So it allows us to essentially turn neurons on and off at our whim. And these are conveniently packaged in a set of vectors that I'm not going to get into that allow us to put them in specific types of neurons in specific places. So what I'm going to show you now is data, what happens if we turn on a GLP-1 receptor neuron in the area postrema, and what happens if we turn on a GLP-1 receptor neuron in the NTS. And what I'm showing you here is the green that's associated with the expression of the DREAD, and then all of these little white dots, that's standing for something called FOS. FOS is basically evidence that the neuron has been firing, immunohistochemical evidence. And I'm telling you about that because you're going to see that again in another context in a minute. So the bottom line is if you give CNO to animals that have DREADs in their area postrema GLP-1 receptor neurons, you can turn them on. And when you do that, food intake goes down by about 50%. I'm just showing you the 4-hour data, and I'll show you additional data later. Same thing in the nucleus of the solitary tract. We can transduce them with DREADs. We can turn them on. That's what all those little white dots are. And again, we can push food intake down by about 50% over 4 hours when we do these experiments. We can actually do the opposite experiment too. Now here we're not using DREADs, we're using tetanus toxin. Tetanus toxin actually doesn't kill the cells, it prevents neurotransmission downstream. They can't release neurotransmitters. So what we're doing here is functionally silencing the neurons. The neurons that we're silencing here are the ones that express GLP-1 receptor that are in the nucleus of the solitary tract. And you can see that that doesn't do much to the body weight of these animals over the first 6 weeks when they're just eating sort of your standard shredded wheat type chow diet. But the second you put them on the McDonald's or Tim Hortons like diet, they start eating like crazy and their body weight goes up by about 50% over the following 6 weeks. And there are the food intake data. So what this says is that the GLP-1 receptor expressing neurons in the NTS actually not only can turn off food intake when you turn them on, but they are actively turning off food intake over the long term in our brains all the time. We haven't actually tried to do this with the area postrema GLP-1 receptor neurons yet because there are a variety of technical issues that I won't get into. So what I've shown you is that area postrema GLP-1 receptor neurons can mediate the suppression of food intake. The NTS GLP-1 receptor neurons can do the same thing. And that the NTS GLP-1 receptor neurons are actually involved in the long-term restraint of food intake and body weight. Now, this is back to that settling point diagram that I showed you earlier, showing that if you have less fat or easier to eat tasty calories, that tends to push up food intake and body weight. If you have decreased fat reserves or harder to get less tasty food, that tends to push things down. And what I've just shown you is that if you push on neurons in the nucleus of the solitary tract that essentially cause early satiation, that also pushes down. But most of you will probably realize that this flies in the face of what I've been telling you since the very beginning of this, which is that hypothalamic systems are the thing that should do this, not brainstem systems. Anytime we decrease the size of a meal by pushing on a brainstem system only, that should be compensated for by eating more meals. And it often is when we push on brainstem systems using other things. So what's different here? So this is back to our diagram of the hypothalamus, again, reminding you about the POMC neurons that say stop eating, the AGRP neurons that say go for it. And I'm going to show you, again, this is FOS data. If you fast an animal overnight, you can see that there's a lot of FOS down here in what we call the medial basal part of the arcuate nucleus. And again, FOS means the neurons are firing. All of this FOS is co-localized with NPY. So what I'm showing you here is that all of this fasting-induced FOS down here is evidence that those AGRP neurons are firing like crazy. So this is a version that doesn't have the NPY GFP, but you can see in a fed mouse, there's very little FOS down there. Again, here's fasting-induced CFOS in the AGRP neurons. If you refeed the animals, not surprisingly, it all goes away. But it turns out that if you turn on a population of neurons in the nucleus of the solitary tract, these turn out to be calcitonin receptor neurons, you can actually turn off the AGRP neurons. So what that means is that there is actually a link between the nucleus of the solitary tract and these brainstem systems, or at least some of them, and the populations of neurons in the hypothalamus that control long-term energy balance. They're not as separated as we thought they were. And of course, that means that the brainstem actually looks like a better target for the control of food intake than we initially thought it was. Okay, let's go back to GLP-1 receptor agonists. And I'm using semaglutide as my example here, but obviously there are lots of GLP-1 receptor agonists out there, and I'm not trying to pick on semaglutide. So what are the problems with it? Why aren't there more people taking it? Access. So it's really hard to get, and it's extraordinarily expensive. So most people can't get their hands on it. But the other thing, and I'm just pulling a quote from the New York Times from one of my colleagues at the University of Michigan, is that this stuff causes nausea and diarrhea and dehydration. Again, it's not just semaglutide, tirzepatide, and other GLP-1 receptor agonists do the same thing. And in fact, this is bad enough that about half the people who start on these drugs discontinue their use within a year. So the question becomes whether it's possible to separate the suppression of eating with GLP-1 receptor agonists from the nausea that they produce. And I'm going to start, just see how I'm doing time-wise. Okay, we'll go. So is it possible to separate the suppression of eating with GLP-1 receptor agonists from nausea? So when I started thinking about this problem like 8 or 9 years ago, I thought about it based on my own experience. And then you think about it, if you eat a little bit of food, at some point in time, you start to feel full. You eat more, maybe it's Thanksgiving dinner, at some point in time, you get really full. And then if you eat even more than that, you become very uncomfortable. My emoji didn't come up. That's supposed to be a barfing emoji. Sorry. In any case, you get sick. And so what this suggested to me in my naivete is that there actually is a single kind of system in the nucleus of the solitary tract. And if you push on it just a little bit, it terminates meals and gives you satiation. But if you push on it too hard, it creates nausea and aversion. And that this would predict that you actually can't separate appetite suppression from nausea. Fortunately, I have a colleague at the University of Michigan named Randy Seeley, who knows a lot more about the communication between the gut and the brain than I did at the time. And he told me that that model didn't make sense. And why it doesn't make sense is the gut has to tell the brain a couple different things. So if you eat food, the gut actually has to tell to the brain that there's actually good food in there. And as a consequence of that, the brain needs to modulate all sorts of things, including gut physiology, to increase the ability to absorb those calories, to create satiation when you've ingested enough calories, and also to create a rewarding sensation that tells you, hey, that was good stuff, you should go back and eat that again. On the other hand, if you ingest bad food, a toxin, an irritant, something that's got E. coli on it, right? Then the gut needs to tell your brain that it's time to stop eating, but it's a completely different response, right? It doesn't, well, you don't want to absorb that stuff. So it says, not only don't stop eating, but expel that stuff, nausea, vomiting, diarrhea, and also don't ever eat that again. It needs to create an aversive memory so that you don't go back and eat things that are poisonous, for instance. And it doesn't make sense to me, and hopefully to you, that one circuit would send both of those signals just based on how hard you pushed on it, right? So I'm going to set this up by telling you that there's a population of neurons in the nucleus of the solitary tract that expresses cholecystokinin, which I know is a gut hormone, but here it's just a neurotransmitter. And if you turn these things on with dreads, you decrease food intake and you get a big aversive response, right? Animals, mice, rats don't throw up, but we have ways of measuring it. And I'll get into that in a minute as well. So we hypothesize that there is another set of neurons in the nucleus of the solitary tract that would mediate a different signal that would basically be able to suppress food intake without causing aversive effects. And I'm not going to go into the details of it, but we decided it was a set of neurons that I'm going to call LC neurons for want of a better term right now. So what I'm showing you here is what happens when we use DREADS to activate either the CCK neurons or the LC neurons. The CCK neurons are in pink, and you can see that we get a nice suppression of food intake over 2 days of CNO treatment. Same thing for body weight. When we activate the LC neurons, actually, we turn off food intake completely over that first 24 hours. The mice don't eat a thing. They eat a little bit that next day, and of course, their body weight goes down. We were originally going to do this experiment for 4 days, but it turns out they had lost so much weight that if we kept going, we would have had to euthanize them, so we stopped it early. So how does this make these animals feel? Again, mice and rats don't vomit, so we have to use kind of funny assays to get them to tell us how they feel. And the assay that we use is something called a conditioned taste aversion assay. Basically, we get them used to drinking out of water bottles, we get them really thirsty, and then we give them a water bottle that has a novel taste in it. In this case, it's saccharin. And at the same time we give them that water bottle that has the flavor in it, we actually turn on a set of neurons in their brain or give them some other stimulus. And if it made them feel bad, then a couple days later, when we do the same thing and we ask, do you want to drink the saccharin in water or just the plain water? They say, hey, I don't want to drink the saccharin. This is why I haven't had tequila in 40 years, right? This is a conditioned taste aversion. Okay, so what happens if you turn on the CCK neurons? So lithium chloride actually causes gastrointestinal malaise, and you can see that it makes the animals really not like drinking saccharin. Same thing when we turn on the CCK neurons. On the other hand, when we do this turning on the LC neurons, it turns out no conditioned taste aversion at all. And that's despite the fact that we were pushing hard enough on these neurons that the animals ate virtually nothing over 2 days. So it is actually possible to separate the circuits that cause suppression of food intake with aversion and the suppression of food intake without aversion. So then the question becomes, how do these GLP-1 receptor neurons fit in? So I'm going to tell you right now something I didn't tell you earlier, which is the area postrema is something called the vomit center in the brain, because there are lots of things you can do in the area postrema that make humans and animals both very sick. And the question is that what is going on with the area postrema GLP-1 receptor neurons? And maybe the ones in the NTS are not quite so bad. So we're back to the DREAD-mediated activation of these things. And it turns out that when you turn on the area postrema GLP-1 receptor neurons, you get a huge conditioned taste aversion. The animals do not like that at all. On the other hand, if you turn on the NTS GLP-1 receptor neurons, the animals are just fine with that. So the GLP-1 receptors in the area postrema cause decreased food intake with aversion. The ones in the nucleus of the solitary tract mediate non-aversive satiation. What does that mean for the control of food intake by liraglutide? So what I'm showing you here is an experiment that we did using one of Randy Seeley's fancy mouse models, where we're looking at a conditioned taste aversion to GLP-1 receptor agonist liraglutide. So you can see that wild type animals, you give them liraglutide and they develop a very strong conditioned taste aversion. If you take animals that have absolutely no GLP-1 receptor anywhere in the body, you give them liraglutide, it doesn't matter, no conditioned taste aversion. But if you turn GLP-1 receptor on in the area postrema and nowhere else, again, these animals hate that stuff. Huge conditioned taste aversion. I'm going to tell you, but not show you, that you don't get a conditioned taste aversion to liraglutide when you turn on GLP-1 receptors in the nucleus of the solitary tract only. But what about food intake? So it turns out that if you take animals and you give them liraglutide, let's see what we have here. We have control animals. We have animals that have no GLP-1 receptor, not surprisingly, no suppression of food intake by liraglutide. If we put GLP-1 receptor in the area postrema only, great suppression of food intake. We put it back in the NTS, nada. And you see the same thing basically when you look at body weight. So what this means is that, yeah, there are sets of neurons in the nucleus of the solitary tract that if you turn them on can give you non-aversive satiation. But it turns out that liraglutide, at least over the time course that we used, doesn't get in there to do anything to them. It's only acting on the area postrema GLP-1 receptor neurons, which gives you nausea and other aversive sensations at the same time that it decreases food intake. So I guess the take-home message here is an ideal drug, which we don't have at this point in time, would turn on those NTS non-aversive satiation neurons, which we know can also turn off the AGRP neurons in the hypothalamus. And we'd like to do that without turning on any of these aversive pathways. If I knew how to do that, I wouldn't be here now. I'd be out making a couple $100 million somewhere, but that's obviously the direction that we're going. So that's all I have to say today, except to actually thank the people who did the work. What's next?
Well, thank you very much, Martin. That was phenomenal. Great story from the beginning to discovery of leptin and all the way through to the GLP-1s that are very popular today and everyone's using them. We only have a couple of minutes, so if there are any questions from the audience first, who's here in the hybrid, we'll take a question. No, if not, then we can ask anybody on the Zoom who might be able to unmute yourselves. There are a couple of comments or questions in the chat, which we can read here. Let me read one. Is the leptin, in the leptin studies in the mice or rats, does the age of the animal matter? Can the rat data be transferred to human models with aging, considering the rats just continue to grow with increasing input? Are the effects the same in children, adults, and the reproductive age? So can you comment on the age effects?
Yeah. So the bottom line is that leptin works pretty much the same way in anybody, no matter their age. It really all is dictated by adiposity. I will say that if you give leptin to a young child, say a leptin deficient young child who's about like 6 or 7 years old, you will not actually push them into menstruation, right? There are other switches in the brain that have to happen to get that to go. But in general, yeah, everybody responds to leptin more or less the same way.
So I had a question regarding the last bit, very, very nice, beautiful work on the aversive response to the GLP-1s. And it seems that that's what mediates all this weight loss in everybody. But there are people who, they say, at least in the pamphlets with the drug, that after 4 to 6 weeks, the nausea resolves and they still lose weight. So can you comment on that?
Right. So again, we don't have data to address that directly, but it is possible to cause prolonged weight loss just by turning on these non-aversive circuits. So even though that's not the way the GLP-1 receptor agonists work, I don't think you actually have to make somebody feel sick to get them to lose weight in the long run. I mean, we won't know that until we have a drug that actually does that, but I think that's the way it's going to turn out.
Great. Let's see, there's first one here. Are there GLP-1 antagonists which may increase body weight in low BMI?
Oh, interesting question. So the answer is yes, there are GLP-1 receptor antagonists. It turns out that they don't do much to body weight unless you put them in to prevent exogenous GLP-1 receptor action. And that's because it doesn't seem like endogenous GLP-1 action in the brain does as much as exogenous sort of pharmacological doses of it do. Now, that doesn't say that the GLP-1 receptor neurons aren't important. It's just that GLP-1 receptor agonist action on them endogenously isn't that important.
Even with the DREAD experiments.
Right.
Are there monogenic mutations in humans in the LC neurons?
So that's a great question. So are there monogenic mutations in the LC neurons in the NTS that can decrease their signaling to the brain? So I can't give you a straight answer, but what I can tell you is we've been working with a really smart, talented bioinformatician who's at the University of Copenhagen, who's very interested in these circuits. His name is Tuna Peres. And what Tuna did is he used single nucleus RNA sequencing to map out all of the different types of neurons in the area postrema and NTS. And then he took the gene expression signatures of each population and he essentially compared that to all of the GWAS identified polymorphisms that track with weight in humans. And based on that, he predicted that the LC neurons would actually be really important for the restraint of body weight. And he actually made that prediction about a couple of different types of neurons in the NTS, which we're in the process of studying.
That's great. Well, we're 1 minute. One last question. Okay. How do you envision targeting the LC neurons?
Yeah. So, boy, I wish I had an answer to that question, because again, if I did, I probably wouldn't be here. I'd be off hawking it to some drug company. So obviously we know the gene expression profile of these things, and there's nothing that sort of stands up and says, hey, target me, right? And the world will be better. But we're trying to map out all of the different populations of neurons in the nucleus of the solitary tract that may mediate non-aversive suppression of food intake. And my hope is that as we map through these things, we'll find one that has, with it, an easy way to target it. But we're obviously just hoping at this point in time.
Okay. Well, we're past the hour, so I'd like to once again thank Dr. Myers for a fantastic talk, and thank Novo Nordisk for sponsoring the lectureship. Thank you.